Well, hi everyone. I'm Adrian, the Inpatient Chief of Medicine here at Montlake. Welcome to our Department of Medicine Grand Rounds. Just a couple of housekeeping things before I turn it over to Dr. Lyles to introduce our speaker today. I anticipate we'll have some time for Q&A at the end. And so if you have questions for Dr. Wu at the end of his talk, please submit them via the chat and I will read them out. And without any further ado beyond that, I will turn things over to Dr. Lyles, our very own Dr. Lyles, to introduce our speaker today.
Okay, welcome everyone. Today's Medicine Grand Rounds is the annual Vollweiler lecture. And Wade Vollweiler was a physician scientist who was the first, or the founding head of the Division of Gastroenterology. And he led that division for 31 years. And so quite a long tenure as a division head. And the Vollweiler lecture involves an invitation to a notable individual, a leader in the field of gastroenterology, and to give the annual lecture. And this year, the Division of Gastroenterology chose Dr. Gary Wu from Penn to be the Vollweiler lecturer. And Dr. Wu is the Ferdinand G. Weisbrod Professor in Gastroenterology in the Perelman School of Medicine at the University of Pennsylvania, and he's the director of the NIH P30 Center for Molecular Studies in Digestive and Liver Disease. And he was the inaugural director and chair of the Scientific Advisory Board for the American Gastroenterological Association Center for Gut Microbiome Research and Education, and is an elected member of both the ASCI and also the AAP. He's also the co-director of the Penn CHOP Microbiome Program, which is a campus-wide initiative to accelerate research in the microbiome across the entire university campus. And his current research programs involve mutualistic interactions between the gut microbiome, their interactions with the host, with a particular focus on metabolism. And he was appointed as the founding director of the Penn Center for Nutritional Sciences and Medicine, a joint collaboration between the Penn School of Medicine and the School of Nursing. And as a physician scientist, he is very interested in the physiology and biochemistry and of the gut and its relevance for human health and disease. He has significant administrative experience in multidisciplinary team research, and he has published numerous senior authored manuscripts in high-impact journals, including Science, Nature Microbiology, Cell Host and Microbe, Gastroenterology, and the Journal of Clinical Investigation. So please welcome Dr. Wu as our 2025 Vollweiler lecture, and I'll turn the podium over to Dr. Wu. Thanks for accepting the invitation.
Great, thank you very much for that generous introduction, and I'm delighted to be here. I'm really honored to give this presentation. So what I thought I would do over the next 45 minutes or so is talk about why the gut microbiome is relevant to human health and disease. I'm really going to sort of emphasize translational aspects rather than focus on sort of wet bench mouse research. So I will mention some of the things that we've learned in a wet bench laboratory, but always within the context of how this is actually relevant to the patients that we ultimately treat. So these are my disclosures, none of which are relevant to my presentation today. So microbiology began about 300 years ago with Antonin von Leeuwenhoek, who described the first microbes using the optics in the left lower hand corner here. But the field of microbiome research has really exploded over the past 20 to 30 years, and it's largely based on technology development, and that is high throughput DNA sequencing that allows us now to sequence billion base pairs of DNA within a very short period of time. And it's for that reason now that we can describe our microbial environment with unprecedented precision in ways that we've never been able to do previously. And so the number of publications has increased exponentially over the past couple of years. So this really means that we've learned a lot in the microbiome space in a relatively short period of time, but it also means it's a relatively new area of science where we've learned a lot, but there's still a lot of things that we need to learn about the microbial environment and how it actually affects us, and specifically how this could be important in the treatment and or prevention of disease. So our microbiomes on our body surfaces are actually very different depending on the body site that you're interested in studying. Most of what we know is actually bacteria, but it's important to know that there are different types of organisms that live on our body surfaces. It's not just bacteria, but there's another complete different domain of life called archaea. These are extremophiles that, for example, in the gut produce methane, so methanogens, microeukaryotes like fungi and yeast. And I'll spend maybe a minute or two at the very end of my talk talking about viruses. So each body site being very distinctive, the gut microbiota is particularly unique because it's one of the most densely populated microbial communities in the biosphere. It's very diverse, containing a lot of different types of microbes. And when you sort of look at the structure of the gut microbiota, it's always very distinct from all the other body sites involved. And that's what I'm really going to focus on in my talk today. So this is that 30,000-foot view of how we interact with the gut microbiota. So the gut microbiota lives in the external world topologically. It's in the lumen of the gut. The internal world contains the epithelium and the mucosal immune system. And what separates us from the external world, the gut microbiota, is a thin layer of mucus, which in the colon is completely sterile and separates us. Nevertheless, we communicate with the gut microbiota because we can produce mucus and we produce antimicrobial peptides that flow into the lumen of the gut, help to fortify barrier function, and can change the functionality and composition of the microbiota. And in return, the microbiota can make small molecules like short-chain fatty acids, polysaccharide A, that can induce immune tolerance and have an anti-inflammatory effect. Other types of microbes can have a pro-inflammatory effect. And bacteria in general can help program plasma cells to secrete specific immunoglobulin A that then is transported into the lumen of the gut, can bind specific bacterial taxa, again, helping to fortify barrier function. For the purposes of my talk, it's important to recognize that what you eat in terms of diet, antibiotics, and xenobiotics not only change the structure of the microbiota, but diet in particular is a substrate by which the microbiota can produce a whole myriad of small molecules that we normally cannot produce. But we can absorb these metabolites that can circulate throughout the body, engage various types of mammalian receptors, and have a significant impact on host physiology. So it is the ability of these microbes to produce these metabolites, as well as alter mucosal immune function, that we believe that the microbiota then can be playing a role in the pathogenesis of many different types of diseases. And I only show you a small snapshot of a number of diseases that have been associated with the gut microbiota. Now, many of these diseases have in common the notion that inflammation is involved. Again, supporting the notion that immune responses to microbes may be important in disease pathogenesis. So I wanted to touch upon 4 different topics very briefly, and this is basically sort of an overview topic on touching on these different topics here. So the first is the gut microbiota as an environmental factor involved in pathogenesis of disease associated with industrialization. I'll then talk about the practical considerations of fecal microbiota transplantation with a note of caution about how we should be doing this. I'll talk about metabolic function of the gut microbiome and why this is relevant in terms of drug therapy. And then again, as I mentioned, I will finish talking a little bit about viruses and why this might be relevant in health and disease. So starting from the very beginning, as infants, we're born sterile. And if you're an infant born into a normal microbial environment, you develop the normal microbiome. That for all the reasons that I showed you on my introductory slide, you develop a normal immune system, which means you have normal immune tolerance. So if you're infected by an organism, you develop an inflammatory response, you get rid of the infection, the inflammation goes away, and you're largely healthy. On the other hand, if you're an infant born into an abnormal microbial world, then our microbial world is actually very different than it was centuries ago. We're living in a much more sanitized environment. We're using antibiotics. Our diets are very different. There are many reasons why our microbial environment is actually very different now. Infants born into this type of world have a different type of microbiota that now may actually program your immune system to be more immune prone, that somewhere later on in life, due to some type of other environmental cofactor, leads you to now respond abnormally to microbial products and antigens, leading to chronic disease like atopic disease, inflammatory bowel disease, metabolic syndrome. So the whole notion is that early life exposure to microbes and diet could actually predispose to development of disease later on in life. So the infant microbiota is actually very dynamic. There are very few things at the very beginning that you're inoculated with, and then after about 2 or 3 years, it becomes very rich and much more stable, looking more like an adult. But in the first 2 years of life, which is shown on this diagram here of a single infant, you can see the colors change very dramatically. And a lot of these changes are timed with significant events in life. The consumption of formula or breastfeeding to solid food, the use of antibiotics, development of fever, all of these things can lead to substantial alterations in infant gut microbiota that may also play a role in immune development. So I'm going to give you one example, and that is the example of atopic disease, in this case, asthma. So I think atopic disease is a really good example of environmental factors that play a role in predisposition and the development of disease. So in this particular study, investigators realized that their asthma was much lower in Amish populations compared to Hutterites. So Amish and Hutterites are genetically very similar to each other, but Amish communities live in a much more agrarian culture than Hutterites, which live in a more industrialized setting. They went into households of children that live in Amish versus Hutterite households, collected house dust, and did sequencing and realized that the microbial environment was somewhat different in those two different environments. They did immune profiling of these children and found there were differences in immune tone and immune profiles. They then closed the loop by taking the house dust they had collected from the households of Amish and Hutterite homes and inoculated mice in an experimental model of allergic asthma, and found that dust extracts from the Amish households, but not the Hutterite households, significantly reduced disease, suggesting that there was something different about the Amish environment and house dust, possibly microbial in nature, that was protective in the development against atopic disease. Something that you think about in the opposite direction in a more of an industrialized setting that is more pro-inflammatory. So I use as an example of this peanut allergies, right? So if you're predisposed to develop peanut allergies, what you don't want to do is keep your child away from peanuts because they could be hypersensitized and develop anaphylactoid reactions to peanuts, antigens. What you do want to do is to expose them safely to low levels of peanut antigen to tolerize the immune system, to reduce hyperreactivity to peanut allergens. The whole idea that the gut microbiome may be fundamentally important in regulating immune tolerance. And somehow our environment has changed and the microbiota is no longer leading to well-established immune tolerance. And that's why we have overreactive immune responses to various types of antigens. So I'll give you an example as a gastroenterologist. When I started practicing over 30 years ago, when I saw an adult patient with eosinophilic esophagitis, that was a case report in an adult. It was so rare. But now we routinely biopsy the esophagus in individuals that have esophageal symptoms on a proton pump inhibitor because we're worried that they have eosinophilic esophagitis because it's so prevalent now. So that really, in even the time period that I've been practicing gastroenterology, there's been an explosion of atopic disease. And we think that obviously that's an environmental factor. So the gut microbiota is very different in different environments. So this is the gut microbiota of children living in rural Africa versus in Europe. And you can just see the colors being very, very different. The gut microbiota is fundamentally different. And so why is it actually different? And it turns out that individuals living in rural Africa have higher microbial diversity, meaning more different types of microorganisms living in the gut, than individuals living in Europe, which have decreased diversity. You have less different types of things in the gut. What's the biological basis of that? Well, in part, it's actually diet and dietary fiber. We know that individuals living in rural Africa consume greater than 50 grams of fiber a day. In industrialized settings, you're lucky if you're consuming 10 to 15 grams of fiber a day, so only a fraction of the dietary fiber that you take. And so there is a notion that dietary fiber could be playing a role in a difference in the composition of microbiota, and that's been demonstrated in a mouse model by Justin Sonnenberg, shown in this diagram down here. So if you have a mouse that is fed a high fiber diet, from generation to generation, they actually maintain the richness of their microbiota. So each generation has all the different types of various organisms from generation to generation. On the other hand, if you have a mouse that is fed a low fiber diet, from generation to generation, you begin to lose more and more microbes, becomes less diverse. And even within one generation, if you go from a low to high fiber diet, you can recover some organisms, but you can't recover all the organisms. So that, in essence, is what is mimicking what we actually see in industrialized nations. We have essentially been losing over time various types of microorganisms, particularly bacteria in the gut, and we now have a less diverse microbiota over time, possibly because we're consuming less dietary fiber. So this is observed in mice. Is this relevant in human biology? Well, Dan Knights has done a study where he looked at the microbiome of individuals emigrating from agrarian cultures in Southeast Asia into the United States in an industrialized settings, and they could actually see the same thing. There's a decrease in diversity of the microbiota upon immigration, and you begin to lose organisms like Prevotella, which is organisms that's very good at digesting dietary fiber. You begin to lose these organisms that they're being displaced by other types of organisms. So there is a concern that over time, as we lose these microbes that normally we have co-evolved with over the millennia, that perhaps they'll be extinguished from the biosphere, because some of these can only live in the gut, are very difficult to live outside the gut. So there are studies ongoing right now trying to preserve some of these unusual organisms with the notion that ultimately they may be important for health, but we don't want them to completely be lost from the biosphere. It's sort of like the seed bank that is actually being developed to sort of preserve for posterity these different types of microbes. So why do we focus on dietary fiber? Well, we live in a mutualistic relationship with the gut microbiota. We produce substances like mucus, which is a source of nutrition for the microbiota. And in return, the microbiota can digest dietary fiber and make beneficial small molecules like short-chain fatty acids. But why is the microbiota so good at digesting these complex dietary fibers or complex glycans? Is because collectively the genome of the microbiome contains many different types of genes called glycoside hydrolases that are very good at digesting really complex carbohydrates in a way that we're not able to digest. And that's why not only can they break down these dietary fibers, but they actually depend on the dietary fiber for their viability. And again, as I mentioned previously, the digestion of these dietary fibers or complex polysaccharides leads to the production of beneficial short-chain fatty acids that can induce immune tolerance, have an anti-inflammatory effect. It can alter the structure of the genome through epigenetic effects, and short-chain fatty acids are a fuel source for the colonic epithelium, helping to fortify barrier function. So I'm going to use inflammatory bowel disease as an example for how we can think about therapeutics by modifying the gut microbiota. So as you know, inflammatory bowel disease can be roughly divided to Crohn's disease, which is inflammation anywhere in a gastrointestinal tract from the mouth to the anus. Ulcerative colitis is really focused on the colon. There are many other phenotypic differences between these two diseases, but the bottom line is once you develop inflammatory bowel disease, you're basically subjected to lifelong immunosuppression without a cure except for a total colectomy if you have ulcerative colitis. The pathogenesis of inflammatory bowel disease is complex. It's partly a genetic predisposition that leads to immunoregulatory defect in response to an environmental trigger. And we think the most important environmental trigger is the gut microbiota and an interaction with diet. So we know that environmental factors are very important in development of inflammatory bowel disease. And this is based on identical twin studies where the concordance of Crohn's disease between identical twins is only about 30 to 40%. And only about 10% for ulcerative colitis. That means the majority of the risk for inflammatory bowel disease is likely to be environmental. And in fact, the incidence of inflammatory bowel disease has rapidly been increasing in Western civilization associated with industrialization. And now with industrialization in Asia, we're seeing a lag phase, but we're actually increasing very rapidly. So these are very, very powerful environmental effects associated with many different types of diseases, including atopic disease, metabolic syndrome. And really the issue is, what are these environmental factors? One of these environmental factors may actually be diet, and I'll touch upon this a little bit more. There are epidemiologic studies that suggest dietary fiber, plant, vegetable intake seem to reduce be associated with the reduced risk for Crohn's disease in particular, less so with ulcerative colitis. There has always been an interest that perhaps the ultra-processed diet, processing of food, could be very important in the pathogenesis of disease. There is epidemiologic evidence that the consumption of ultra-processed foods that contain dietary additives is associated with the increased risk, again, for Crohn's disease, not so much for ulcerative colitis. Now, investigators have been studying over the years different types of food additives, dietary emulsifiers, artificial sweeteners, food colorings, nanoparticles, and each one of these has been associated with a change in the microbiota, a decrease in diversity, and other types of mechanisms by which, through the microbiota, you lead to an inflammatory response. I'll talk briefly about dietary emulsifiers per se, because it's one of the things that we're most interested in. And essentially, the consumption of these dietary emulsifiers allows the penetration of bacteria into the mucus, getting closer to the intestinal epithelium, leading to an inflammatory response. Now, the whole notion that the gut microbiota could be involved in the pathogenesis of inflammatory bowel disease is based on this observation here. The gut microbiota in inflammatory bowel disease is very different than in health. And you can see that by the colors. So the appearance of this yellow color here are proteobacteria. These are Enterobacteriaceae. These are E. coli and Klebsiella. They seem to be overrepresented in inflammatory bowel disease. And that's the notion of dysbiosis, or an altered structure of the microbiota associated with the disease process. Why does it look different? Why is it different? It's because it's a complex microbial community that is responding to a difference or a change in the environment. And a change in environment in inflammatory bowel disease is inflammation. So inflammation is leads to an oxidative environment. And these Enterobacteriaceae, these proteobacteria, are oxygen-tolerant organisms. The inflammatory process can also produce small molecules that promote anaerobic respiration. And anaerobic respiration would also lead to the outgrowth of these Enterobacteriaceae. So we cause that dysbiosis. Intestinal inflammation causes this difference in structure. But the question is this difference in the structure of the microbiome, this dysbiosis, does that play a role in perpetuating disease? Well, we think that it probably does, because in animal models, you can take the fecal material from a mouse that has colitis, transfer it into to a healthy wild-type mouse, and under circumstances, that mouse will now develop colitis. So that transferability of disease phenotype simply by transferring the microbiota suggests that the microbiota could be playing a cause and effect role in the development of disease. That's in mice. Do we have evidence in humans? And I'll show you that in a minute. This is important because if dysbiosis is associated with disease, maybe you could change that dysbiotic microbiota in a way that would promote health as a therapeutic option. And there are many different ways that you might be able to do this: probiotics, prebiotics, antibiotics, fecal transplantation, dietary interventions, and I'll touch upon a number of those different things. So as proof of concept that microbes, bacteria play a role in the pathogenesis of inflammatory bowel disease, we use the example of fecal microbiota transplantation. So as you know, fecal microbiota transplantation, taking fecal material from a healthy individual, inoculating somebody that has disease, either through a nasogastric tube, through a capsule, or through a colonoscope, can be very effective in the treatment of Clostridium difficile infection. But now there are several placebo-controlled studies essentially showing that fecal transplantation can lead to an improvement in disease activity, specifically in ulcerative colitis. Now, I still think that this is experimental. I'm not an advocate for saying that this is a standard treatment for ulcerative colitis, but it's proof of concept that the gut microbiota does play a role in the pathogenesis of inflammatory bowel disease, and it could actually be a target for the treatment of disease. So how would you think about targeting a therapeutic through the gut microbiota for treating disease? Well, the concept is on this slide here. So normally you have firmicutes, which are gram-positive organisms, and these are the gram-positive organisms associated with health that produce short-chain fatty acids that have an anti-inflammatory, immune-tolerant effect. In dysbiosis, in inflammatory bowel disease, you lose the firmicutes and you get these proteobacteria that I talked about before. So the simple-minded approach is, well, if you lose these beneficial organisms, why don't we make a product that contains these organisms and just give it back to somebody that has ulcerative colitis? And the cool thing about firmicutes is these are sporulating organisms. So even though they're obligate anaerobes and they die in the presence of oxygen, you can make them turn into spores, and spores are very resistant to oxygen. So you can actually make a capsule of these sporulating organisms that you can put on a shelf, and you can actually have patients swallow them. And so a product was actually made. We're in a phase 1b clinical trial in patients with ulcerative colitis. They received these capsules. They could show that these organisms actually engrafted, and there was an early signal that perhaps it was providing some benefit to these patients with ulcerative colitis. That's a phase 1b study, but unfortunately in a phase 2b study, this strategy failed. They didn't meet their primary endpoint. So what does that essentially mean? That essentially means that treatment of disease in a mouse sometimes does not translate to the effective treatment in humans. It does also mean that further work is actually needed to actually come up with therapeutics for inflammatory bowel disease, focus on the microbiome. But I think investigators learned a lot from this experience. And I think that there are newer, more sophisticated strategies that are online now to be thinking about better therapeutics using the microbiome for the treatment of inflammatory bowel disease. Again, this is a very new area of research. It's only been around for 20 to 30 years. So there's more work that needs to be done. So let me talk about fecal transplantation. So Clostridium difficile infection, as you know, is a huge problem. It's growing in incidence, at least a significant amount of morbidity and mortality. The reason you get Clostridium difficile infection is you take an antibiotic, you disrupt your microbiota, it makes it susceptible, that allows C. difficile spores to germinate. Once they germinate and they turn into vegetative organisms, they produce a toxin that then leads to development of colitis. And there's a whole spectrum of disease activity, everything from asymptomatic carriage all the way to death. So there are high recurrence rates of Clostridium difficile infection, making them refractory to treatment. and about 20% of individuals that don't respond to current standard treatments with antibiotics. And so the therapeutic opportunity is actually to treat this susceptible microbiota. And the most effective way to treat that susceptible microbiota, again, is through fecal transplantation that actually targets that dysbiotic microbiota to restore the complexity and diversity of the microbiota, preventing the growth and sporulation and germination of C. difficile. Highly effective, leads to a success rate of about 80 to 90% in patients that have refractory or recurrent Clostridioides difficile infection. With the notion that it actually works so well for the treatment of this infectious disease, over a decade ago, I made this slide, and most of the clinical trials were focused on Clostridioides difficile infection, but you saw people beginning to use FMT for the treatment potentially of other types of disorders. But I did the same type of analysis about 2 years ago, and it's actually blossomed a number of clinical trials on clinical.trials.gov across the world, looking at many different types of diseases, potentially targeting disease with fecal transplantation has exploded to over 400 now. So again, it's a very active area of investigation. Let me give you one example, which is a non-infectious disease process where fecal transplantation as proof of concept provided some evidence for validity of a therapeutic approach. So this is checkpoint inhibitor blockade, so PD-1 blockade, the treatment of malignant melanoma. So investigators studied the microbiome of individuals that were non-responders to PD-1 blockade versus those individuals that were responders, and actually could find a significant difference in the composition of the microbiota. They took the fecal samples from responders and non-responders, put them into notobotic mice that actually had a tumor, then treat them with PD-1 blockade, then they could show that the mice that received the responder microbiota actually had a better response in terms of anti-tumor effects of PD-1 blockade compared to the non-responder recipient mice. And this all had to do with immune tolerance. So the non-responders had more immune tolerance than the responders, okay? So this is in mice. The transferability suggests that the difference in the composition of the human microbiota may have a functional effect. And so there are studies that were done using fecal transplantation and back-to-back papers published in Science showing the fecal microbiota transplantation improved refractory patients that were refractory to immunotherapy using PD-1 blockade. So it's proof of concept that changing the gut microbiota could improve outcomes in cancer therapy focused on immune modulation. So now there's a large amount of interest in targeting cancer therapeutics through the microbiota. Now, there's been a lot of interest in the lay public about fecal transplantation, even a video of how you can do your own fecal transplantation. And, when this field first started, there was a lot of concern that we could be passing on pathogens like HIV and HCV that we transmitted through blood transfusions. We know that the gut microbiota is very poorly characterized, it's very complex. A lot of animal model data suggesting the gut microbiota could be associated with disease. And so back over a decade ago, there's a very significant concern by the FDA that there should be some type of regulation. However, they had to strike a very significant balance in that if they basically shut down fecal transplantation, all those individuals that have recurrent or refractory Clostridioides difficile infection would not be able to get fecal transplantation for the treatment of disease. And for that reason, they that had discretionary oversight, which meant that only for recurrent Clostridioides difficile infection could you treat without an IND. But this has evolved now because there are new products that are FDA approved. So biobanks like OpenBiome can no longer provide material unless they actually have an IND. So now we're at a very delicate stage. We're trying to balance FMT from biobanks versus currently available FDA-approved products. Now, it is true that there are some examples of pathogen transmission antibiotic-resistant organisms that were transferred, pathogens that were transferred by FMT, and there's a high-profile death that actually occurred with an antibiotic-resistant organism. So it is important that we continue to be diligent and cautious about this. The other way to think about this is that you have grown up with your own microbiota. It's very personalized. Everybody has their own microbiota. When you do a fecal transplantation, you've largely swapped out maybe 20 to 40% of your microbes from the donor. So now you have somebody else's microbes in your gut. As far as we can tell, they're actually there for a very long period of time. Does that mean that perhaps that you could be predisposed to some type of disease later on in life? We don't really know the answer to that, but that is a concern. And for that reason, the American Gastroenterological Association does have an FMT national registry to address all of these concerns by tracking people long-term, understanding how FMT is done in a practical way across the United States. This registry has a lot of different partners and partner organization. It's now collected material information about 1,000 different individuals across the United States over the past couple of years. We have longitudinal follow-up going out to about 2 years with the intent of following individuals for 10 years. And we think that this will have a significant impact on the field, just having this information. Fortunately, so far, and this has been published about 2 years ago, we don't have a significant warning signal. So for at least the first year or two after fecal transplantation, there's not an imbalance compared to control, suggesting there's something dangerous that's going on. We'll just have to wait for longer term follow-up. And nobody would be happier than me than if we were to say, well, there was never anything to worry about, but we would always be faltered if we didn't do our best to try to collect this data to safeguard our patients that are getting these undefined treatments for Clostridium difficile infection. So in the field, we began with FMT. There are processed fecal products, and ultimately we'll get to define microbial consortia, specific microorganisms that you grow in a laboratory, that you combine together. You can think about as the next generation of probiotics. For Clostridium difficile infection, these two first products actually do work. And there's a hope that defined microbial consortia, very specific organisms, when put together as a concoction, could actually be effective in the treatment of Clostridium difficile infection. For the treatment of other type of diseases, this is really the holy grail. Identifying using your best science, specifically what type of microbes provide a benefit, targeting a specific type of disease for the treatment. And the notion is, as we better define what we put into our patients in terms of microbes, the safer it's going to be. So for initiation, we began with FMT, as I showed you in numerous examples. But for sustainability, we do need to be walking down this axis here to get to more sophisticated products that are based on science. So the FDA now has a designation for these new types of products to be approved for the use for the treatment of disease. So it's important to note that there is no probiotic currently on the market that is been approved by the FDA for the treatment and or prevention of disease. Standard probiotics that you would buy as a dietary supplement. But by FDA terms, the live biotherapy product contains live microorganisms such as bacteria, prevents, treats, or cures a disease or conditions in humans since it's not a vaccine. However, there are two now approved live biotherapeutic products. One is an enema, and the other one is a capsule. Both are used to treat Clostridium difficile infection. So even though the microbiome space is relatively new, been around for only about 2 decades, we're now beginning to see new therapeutic agents for the treatment of disease through the gut microbiota. But this extends well beyond just a fecal transplantation and the new LBPs for Clostridium difficile infection. There are a number of different types of disease entities: hepatic encephalopathy, cancer, therapeutics, graft versus host disease. These are in various stages of clinical trials in humans. So we are cautiously optimistic over the next 5 to 10 years that we will see other products focused on the microbiota that could be used for the treatment of a number of different types of diseases, in addition to just an infectious disease like Clostridioides difficile infection. All right, so let me just talk briefly about the gut microbiome and drug therapy. So as I mentioned, what you eat in terms of diet can be converted into small molecules that actually we absorb after first pass metabolism through the liver. It can circulate widely throughout the body. So one example of diet leading to the production of small molecules is a dietary amino acids. So amino acids can be converted into small molecules that have many different types of biological effects. And I'll give you one example of this, and this is aromatic amino acids. So tryptophan being one aromatic amino acid that can be converted through aromatic amino acid reductases into a small molecule called endopropionic acid. We do not make endopropionic acid, but we can readily measure endopropionic acid in the urine and in the blood, but that's from the gut microbiota. Endopropionic acid is a ligand that can activate the PXR receptor, the pregnane X receptor. Activation of pregnane X receptor helps to fortify intestinal barrier function, and PXR is a bona fide genetic locus for the risk of inflammatory bowel disease. Here's another example. So tyrosine, another aromatic amino acid, can be converted by the gut microbiota into P-cresol. P-cresol can be sulfated by the liver, and P-cresol sulfate is associated with increased mortality in individuals with end-stage kidney disease. And the reason for this is not completely well understood, but it may have something to do with lymphocyte function. So the production of P-cresol through the gut microbiota, through metabolism of tyrosine, actually can have effect on acetaminophen or Tylenol metabolism through phase 2 hepatic metabolism. As you know, xenobiotics are often conjugated to either glucuronide, glucuronic acid, or sulfur that allows it to be excreted from the body. But if you have a microbiota that produces high levels of P-cresol, you will actually shift acetaminophen metabolism away from sulfation into glucuronidation. So your gut microbiota can actually change the way drugs are actually metabolized. So getting back to that glucuronidated metabolism, I'm going to give you one example by which the gut microbiota could be disadvantageous. So this is work by Matt Redenbaugh. There's a chemotherapeutic agent called irinotecan that can lead to GI toxicity. So irinotecan undergoes glucuronidation in the liver by phase 2 hepatic metabolism using UDP glucuronotransferase. When you tag a xenobiotic with a sugar like glucuronide, it increases its aqueous solubility and allows it to be excreted in the urine or in the bile. Glucuronidated irinotecan excreted into the lumen of the gut through bile can then actually be reactivated by the gut microbiota, because the gut microbiota actually contains beta-glucuronidases that will take off the sugar. When you take off the sugar, you reactivate the irinotecan, and that can lead to GI toxicity. So it's a demonstration of how drug metabolism could actually be altered through the gut microbiota that would increase toxicity of a particular drug. And there's a whole bunch of different examples now of how the microbiota can metabolize drugs in a way that decreases their activity, leads to toxic metabolites. Levodopa, used to treat Parkinson's disease, is another drug that's metabolized by the gut microbiota into a toxic metabolite, decreasing efficacy. So I think we're beginning to see more and more evidence that the gut microbiota changes the efficacy of our current drug treatments, sometimes leading to adverse outcomes. I'm going to show you one final example of this. As a gastroenterologist, we're very interested in mesalamine, 5-ASA, as a treatment for ulcerative colitis. It turns out that acylation by the gut microbiota of 5-ASA actually inactivates mesalamine, making it no longer effective. Scientists have identified the genes in the microbiota involved in the acylation of 5-ASA. So by sequencing and characterizing the gut microbiota and the genes involved in acylation, these investigators showed that you could predict whether or not somebody with ulcerative colitis may or may not respond to mesalamine based on whether or not the gut microbiota is able to inactivate mesalamine. So it's a whole notion of eventually precision targeting therapeutics by looking at the structure of the gut microbiota. The final thing I have to say is here on this slide, and that is that the gut microbiota makes a huge number of metabolites. So when we do mass spectrometry on a fecal sample, we can annotate maybe 1,000 or 2,000 small molecules, but there are 50 to 70,000 spectral features. Many of these are small molecules which we don't understand what their structure actually is, but they may have bioactivity. So they're investigators actually taking strains of different bacteria, growing them in culture, taking the supernatant, and then screening receptor libraries for determining whether or not they have bioactivity against human receptors. And with the notion that the gut microbiota may be the source for the next generation of small molecule drugs that could be used for treatment of disease. And the field of natural bioactive discovery has been out there for a long time because organisms spend a lot of energy making these different complex molecules because they have biological activity. And the gut microbiota may be the source of certain types of molecules that might be very valuable in the future. So the final thing, let me just touch upon viruses. So viruses, so there are 3 domains of life. And I think viruses are generally regarded as non-living entities, but they are amongst the most successful entities in the biosphere numerically. Enormous, 10 to the 31 viral particles on planet Earth, enormous numbers. And most of these viruses are called bacteriophages. These are viruses that infect bacteria. So bacteriophage have 2 different life cycles. They can infect bacteria and incorporate their genome into the genome of bacteria. And this is called the lysogenic cycle. And lysogeny leads to different functionality because the genes from the phage have now been incorporated into the bacterial chromosome that are now perpetuated. So we think about, for example, Shiga toxin and cholera toxin as being bacterial in origin. Well, in fact, those toxins actually originally came from phage that incorporated that toxin into the genome of bacteria. But there's another life cycle called the lytic life cycle, where bacteriophage will actually infect the bacterium. It'll populate the bacteria, will kill the bacteria, will lyse open the bacteria, and they go on to infect other bacteria. All right, and that lytic phase is something that has been of interest in Eastern European communities for a long time, thinking about phage therapy. And now investigators have taken this to the next level now, where investigators have now, for example, developed phage cocktails to target specific bacteria that are associated with inflammatory bowel disease. This is another story by UCSD, Bernd Schnabel, who has actually targeted Enterococcus, which is associated with severe alcoholic liver disease as a treatment for alcoholic liver disease. There is a clinical trial ongoing right now for phage therapy in a treatment of cystic fibrosis. And just recent release is the Human Virome program, which now is a $170 million project where they're actually mapping the entire virome in human population. So a big area of interest. I think we're going to be seeing a lot more of this in the future. So the microbiome is very complex, trans-kingdom, a lot of different types of organisms that interact in many different ways that can be playing a role in both health and disease. So my last slide is here. When we began doing microbiome research, maybe over a decade ago, we had a lot of animal model data showing functionality. We had human association studies, but at the end of the day, we want to get down here to new diagnostics and new therapeutics. We see the FDA has a new designation for live biotherapeutic products, a proof of concept for reduction to practice and treatment of disease is FMT for Clostridioides difficile infection. So we need to be moving down this axis. And 10 years ago, when I sort of made this slide, I put an arrow here saying this is what we need in the field to be doing more human intervention studies. And now it's really happening. As I showed you on the slide, there are clinical trials now ongoing for the next generation of therapeutics using the microbiome. And it's a beautiful intersection between science and technology, clinical practice that we have FMT and new treatments for Clostridioides difficile infection. Regulation I touched upon a little bit in terms of the FDA. There's a lot of intellectual property and business opportunities. There have been several $billion that have been invested into the microbiome space. So challenging, a lot of really interesting science, but I think we're beginning to see reduction to practice with a new generation of therapeutic products. So with that, thank you very much for allowing me to give you this presentation, and I'm happy to answer any questions if you have any.
Thank you so much, Doctor. That is a profoundly fascinating lecture and topic, and had me in regretting the donut I had this morning, I will say. I want to open it up to questions. And I actually did have one as well, which was several, but one I think later in the talk you mentioned that there's some hope for kind of trials specifically targeting atopic disease processes, and I'm wondering, I guess that sort of implies to my mind the like immunogenic effects of the dysbiosis are reversible, and that there's hope that you can reverse kind of whatever changes are happening in the immune system such that the kind of like pro-inflammatory or autoimmune processes that result are themselves reversible. And I'm wondering how you think about that, and like is the kind of are the effects of dysbiosis fixed early in life, or are they more plastic than that?
Yeah, I think there's a lot of plasticity, a lot of things that we still don't understand. So I'll give you one example. So breastfeeding has been shown epidemiologically to be associated with a decreased risk for atopic disease compared to formula feeding. The same thing we actually see with cesarean sections. The cesarean section is associated with a greater incidence of atopic disease than vaginal birth. Those two entities, the type of feeding and vaginal versus cesarean section, are also associated with a change in the microbiota. So there are notions that exposure early on in life. There are studies ongoing using a vaginal swab, a vaginal sponge to swab your infant that's born by C-section to repopulate with the normal vaginal microbiota, with the notion that you might be able to induce immune tolerance or prevent the development of disease. Whether or not later on in life, what actually happened to you early on is reversible, I don't know the answer to that. I think we're still at the stage now where we have to convince ourselves that these early life exposures could be playing a role through the gut microbiota for the predilection of disease. It makes a lot of sense. Animal models suggest that is the case. Epidemiologic associations suggest that is the case, but we don't have like the smoking gun that essentially says that alteration of microbiota is the cause of a particular disease process. Having said that, my notion, it probably does play a role, but in my mind as a physician, it's all about effect size. It could be that it does play a role, but the size is so small relative to all other types of things that could be playing a role in the pathogenesis of disease, it's almost inconsequential. However, I use the notion of immune tolerance and food allergy as an example where we know that you can induce immune tolerance by exposure to low levels of antigen. That's a real thing. That's cause and effect. That is in clinical practice now. The issue is how much of that extends to the gut microbiota, and I think that that's for the future. I don't think we have strong things that we can do right now. So I hope that answers the question. It's essentially saying it's promising. We do have some evidence. We don't know the effect size. And the reversibility is the next step in the next question. I think we still have to answer the first question.
Yeah, thank you. I think that does answer the question. I appreciate it. And I mean, I guess you would a corollary to that is thinking about the Hutterite and Amish distinction, and like that would imply a fairly large effect size given the differences, but hard to say in other populations.
Yeah, I mean, the effect size was pretty profound in an animal model system. Obviously, I chose the asthma because I think atopic disease is a really good example of immune tolerance. And immune tolerance is, one of those things in common with inflammatory disease responses and inflammatory bowel disease, even immune tolerance being bad in checkpoint blockade therapeutics, right? So I think a lot of this, I didn't really point out graft versus host disease. So actually, there are phase 3 clinical trials right now on modifying the gut microbiota to decrease the risk of graft versus host disease. And that actually makes a lot of sense because, graft versus host disease, you're basically taking a naive or reintroduced immune system that is now being exposed again to a different type of environment. And it does make a lot of sense to me that the microbial environment could be changing immune tone in a way that could be favorable or disfavorable in development of graft versus host disease. And I think the data is fairly convincing. People have been studying this for a decade now, and I think they're getting closer to be thinking about what type of products could be used to decrease the risk for and severity of graft versus host disease.
Wonderful. Thank you. A question from the audience. If you could please provide a comment on the common bacteria that are added to unsweetened yogurt.
Yeah. So as you can see on my disclosures, I do full disclosure. I'm on one of the boards for Danone, and so they add lactobacillus and bifidobacteria to yogurt. So these are organisms that are really important in milk fermentation products. They make the products actually taste good. Do they have a health benefit? Well, bifidobacteria are thought to be uh, beneficial, especially early on in life, because breast milk, as you know, contains human milk oligosaccharides and leads to predominance of bifidobacteria. So there are many reasons to believe that it's beneficial. Um, I would say that, and there is, I think, some evidence that suggests that consuming yogurt seems to be associated with decreased risk of metabolic syndrome. Effect size is not large, but it is sort of somewhat of an association. So I think the food industry has been all over these probiotics that are actually there. None of them, as I reiterated before, reached the bar for FDA approval for the treatment or reduction of risk of any different type of disease. I don't think that they're deleterious in any way. And if you like the taste of yogurt and things like that, I think that's awesome. That's good. I don't think it's going to hurt you. And I could say that, realistically, does it provide some benefit? Maybe it does. It's just that the effect size in human clinical trials doesn't meet the bar for approval from the FDA standpoint as a medical therapeutic.
Great. Thank you. We'll give it another 10 to 15 seconds for any burning questions from the audience. And otherwise, yeah, thank you so much for joining us today, Dr. Wu. We really appreciate you sharing your expertise with us. And this is seems like we are standing on the precipice of some pretty remarkable breakthroughs in this field. So I really appreciate you sharing your expertise with us today. I hope the rest of your visit and time with family is enjoyable and that we give you some unrepresentative Seattle weather to enjoy. And thank you so much for joining us today.
Yes, my pleasure. Thank you very much for the invitation. Really appreciate it.
Bye. All have a great weekend. Thank
Okay,
you.
bye-bye now.